animal imaging instrumentation application. I'm currently in King Hamad University Hospital. The topic is not relevant to this hospital, but it's actually my previous work in Imperial College London and M.wmv. So as an out for my talk today, some facts are going to be discussed with you. So what small animal imaging can offer to you, why the mouse is commonly used in research, mouse versus human genome, some comparison between these two, comparison of imaging technologies available, functional versus structure, resolution and sensitivity in preclinical as well as in clinical aspects, and PET imaging, animal handling and logistics, some factors or variables that should be taken into consideration like temperature, anesthesia, and then at the end we are going to talk about briefly on some imaging modalities like micro-PET, micro-CT, micro-SPECT, micro-PET, optical imaging, as well as photoacoustic or photoacoustic imaging. Rodents actually have become the animal model of choice for modern biomedical research. 95% of all lab animals are mice and rats. But there is actually some concern because we need to have a lot of animals for each experiment, effects of a drug, biodistribution, pharmacokinetics, and pharmacodynamics, and autoradiography. Conventional methods at the moment actually lack a comprehensive description of spatiotemporal characterization of biological or molecular processes. And also there is an increasing strain of ethical and regulatory issues to minimize the number of animals in research. So what small animal imaging can offer for researchers and scientists, actually in vivo characterizations of new diagnostic biomarkers or new therapeutic agents, and this allows for routine interaction investigations, in vivo ligand target binding affinity, and gene therapy, stem cell tracking, monitoring of the endogenous gene expression, screening of transgenic animals, integrated biology or risk stratification, and real-time imaging of early molecular or genetic changes. Small animal imaging has become a new microscope for scientists to look at living biology. So it helps a lot in drug discovery and development, or what's called a translational medicine. Small animal imaging give you an opportunity for longitudinal studies using the same animal as its own control, which is not applicable in humans, reduction of inter-subject variations, and also reduction and cost of animals, number of animals during the study. It also avoids animal dissection and complicated autoradiographic procedures. Other features for small animal imaging, it's more efficient than clinical counterparts, and also approximately 1/3 of cost. Benchtop optical system as well as ultrasound can be fit in a small laboratory, and also it requires less space. Some limitations for small animal imaging, MRI machines, it needs cost, magnetic shielding is required, and high level of expertise, in addition to lengthy acquisition times. In PET imaging, you need an on-site cyclotron as well as high level of expertise as well in radiochemistry. Drug discovery and development actually is a very long process. It takes approximately from 10 to 15 years to come up with a new drug. $800 million is needed at minimum to come with a new drug and release it into the market. If you are going to start by 5,000 compounds, you are going to end at the end of 10 or 15 years by only one compound. So it's a very long process and time-consuming, and also it costs a lot of money. And the process actually starts first by people who are working in cellular and molecular biology to identify and validate a drug, and this drug needs a suitable ligand to be targeted, and then you need to go through a number of experimental and research work to validate this combination between the target and ligand. Actually, small animal imaging here plays an important role because it gives you an opportunity to look at all these experimental variables like physical properties, in vivo metabolism, as well as drug-drug interactions, pharmacokinetics and pharmacodynamics as well. And some additional studies can be also undertaken like autoradiography and toxicology. And at the end of these cycles, you are going to come up with a candidate, new drug. Then a file is sent to FDA as investigational new drug for approval. And once this application has been approved, you are going to start them by clinical trials, phase 1, phase 2, phase 3, and you end this, at the end of this cycle, you are going to end with this clinical approval for a drug. So while the mouse is commonly used in research, actually mice are genetically very similar to humans. It reproduces very rapidly. It has short lifespans, inexpensive and easy to handle, can be genetically manipulated at the molecular level. Let us compare now between mouse and the human. And you will see for estimated number of base pairs in human is 3.2 billion base pair, but in mouse it's 2.6 billion only. The gene number is 25,000 in human, but it's the same number in mouse as well. So average gene density is 100,000 bases in both species, and the chromosome number in human is 46, but in mouse is 40. So what imaging modalities actually are available for small animal imaging? It's mainly functional versus structural. The functional ones are SPECT, PET, optical autoradiography, but structural ones are MRI, CT, and ultrasound. We can find now some systems, SPECT MRI. Now there's one prototype in Johns Hopkins are producing using cadmium zinc telluride, and I think in the near future we're going to have a commercial design for microSPECT, micro MRI. SPECT CT is now available. Some systems provide this option. PET MRI also are in the near future. In the next few years, we are going to have some micro PET MRI available in the market. PET CT and optical CT and optical MRI are also emerging in the field. Structural functional modality like optoacoustic, we are going to talk about this later on. So resolution and sensitivity are two important performance measures actually for any diagnostic modality. Regarding sensitivity, you can find that optical and radionuclide techniques are the best in characterizing the function, the molecular concentration of the drop. So optical techniques actually can go down to femtomolar or nanomolar concentration, radionuclide in the pico-nanomolar concentration, but MR, ultrasound, CT are coming down. On the other hand, we can find the resolution of these imaging modalities like MR and CT are best in characterizing the anatomical or function or morphological structures, but SPECT and PET optical come behind. So if we are going to compare between preclinical PET and SPECT in clinical as well as in preclinical setting, we will find preclinical SPECT and preclinical PET are best in spatial resolution. The preclinical SPECT imaging now can achieve sub-half millimeter resolution, and preclinical PET actually in the range of 1 to 2 millimeter. Regarding sensitivity, you will find preclinical PET is the best in sensitivity, and the preclinical SPECT come after clinical PET due to perimeter limitation and restrictions during count correlation. Animal handling and logistics, a lot of experimental variables actually need to be taken into consideration, like anesthesia, induction and maintenance, temperature control, physiological monitoring like cardiac and respiratory systems need to be monitored very well, catheterization as well as immobilization. Anesthetics is very important to restrain the animal. And type of anesthetics available are intraperitoneal, intramuscular, as well as inhalation. The most commonly used now approach is isoflurane, and allows you to control the degree of inhalation during the study, but one of the drawbacks is training and the cost. Mouse strains have varying sensitivities to anesthesia and alter the CNS as well as cardiovascular signal, and they can greatly influence the outcome. We are going to review some of these imaging modalities available in the market. Actually, micro-MRI is a 3D structural imaging technique. It depends on proton density, magnetic field, as well as frequency pulse to elicit a relaxation time expressed as T1 and T2. Signal variables depends mainly on density of the nuclei, relaxation times, and magnetic homogeneity of the environment, as well as the rate of transport of molecules within the medium. High spatial resolution is the range of 50 micro or even better. Some micro MRI now provides these high resolution images, especially at high magnetic field, 7, 9.4 and 11 Tesla, or even higher. Higher for isolated organs and MR microscopy. Better spatial resolution allows for better chemical species identification and quantification using MRS or magnetic resonance spectroscopy. MRI provides superior soft tissue contrast with flexible operating parameters, and the more emphasis nowadays are placed on micro-PET, micro-MRI. Micro-MRI has flexible acquisition parameters and different sequences, like it can be used in imaging, microscopy, spectroscopy, functional MRI, and dynamic contrast enhanced MRI, and diffusion weighted MRI. It also helps a lot in drug development, in phenotyping, vascular therapy, cellular metabolism, pharmacokinetics, cell tracking, gene expression, drug delivery, and receptor-based imaging. There is actually a number of micro MRI systems available in the market, like Varian, and also Varian, Agilent, and Bruker bioscans with different magnetic field strengths. The high soft tissue contrast provided by MRI give you a very good image for the brain. These images acquired for a rat brain using one of the available systems in the market is using T1 flash, T2 flash images of protein density. We can appreciate actually the cerebral structures and how we can differentiate between these different parts of the brain. Also, one can appreciate here a cardiac muscle can be delineated very well, and the cardiac chambers. Micro-CT computed tomography is also a 3D structural imaging technique. High spatial resolution can go down to 10 micrometer. Soft tissue contrast is very low, but bone imaging is very high. Angiography and digital subtraction is available now with some micro-CT systems. Dynamic perfusion is very limited to very few systems. Dual CT gating, respiratory and cardiac also can be acquired, and has become necessary now micro-CT and hybrid device like HCT and SPECT-CT and optical CT. One of the concerns actually with micro-CT is radiation dose delivered to the animal. As Dr. Osman mentioned in his still, SPECT-CT has also some limitation in delivering a very low dose to the patient. Also in animals, there is some concern about this. Careful design of longitudinal studies should be undertaken. Also tissue contrast, so you need to inject the contrast during some of the studies. Mainly it reveals structural information related about tissue function. Here is the image we acquired with 50 micrometer using dual gating. And here you can see the pulmonary vessels as well as the cardiac and the cardiac chambers and the cardiac muscles. The pulmonary vessels are illustrated here. Descending and ascending aorta and this bifurcation as well. Here you can see the bifurcation. And left ventricle, right ventricle and the atria. Microspect imaging provides high spatial resolution images less than half sub-millimeter in range. Dual isotope injection is possible with microspect, low cost in comparison to PET. Many of the SPECT clinical compounds are commercially available. FDA approved for many SPECT compounds, relatively long half-life of the most SPECT compounds permits labeling with molecules of slow kinetics like antibodies and proteins. Clinical experience, the clinical nuclear medicine is widely spread and of increasing interest. Detection efficiency is one of the limitations actually and is often is not high enough, and thus higher injected dose or longer acquisition time need to be adopted. These images were acquired using 1,740 megabecquerel full time and full scan, and one at the bottom is 10% of the dose or 10% of the acquisition time. The one lower is 1% of the injected dose or the acquisition time, and as you can see here, the image gets noisier as we go down with dose and acquisition time. Gated acquisition is also possible in this imaging modality. Molecular SPECT imaging has been recently reviewed by our group, and we address the current position of the molecular SPECT imaging among other imaging techniques. We describe the strengths and the weaknesses of this approach, differences between SPECT and PET, and focusing on different SPECT designs and detection systems. Also, some pharmaceuticals of clinical as well as preclinical interest has been described. In the same review, we have found that a number of commercial designs has been adopted, and we can find the MyLabs, which is a 3 gamma camera using a specialized geometry, which is focused on the whole body different collimators for rat and mice. Gamma Medica is a tri-modality platform. We're using cadmium zinc toluene in the SPECT module. NanoSPECT is 4 gamma camera with patent pinhole geometry. The one at the bottom actually is Siemens. It's a tri-modality platform using SPECT and PET and CT. The SPECT and CT are co-aligned on the same axis and PET at the back of the gantry. Here we can see the PET of the system and SPECT CT are co-aligned on the same axis. So the gantry of the CT and SPECT are moving together. Here, one of the features provided by SPECT, you can inject 2 tracers at the same time. These 2 tracers, for example, this one is lutetium-177 red is a common. Is a control peptide, indium-111 RGD peptide, small peptide that binds with high affinity to integrins or alpha-v beta-3. And the tumor model used here is glioblastoma, astrocytoma. We can see the selectivity of uptake here for indium-labeled RGD when compared to the number of control red lutetium-177. The magnification provided by the pinhole geometry actually allows to improve the resolution of the images, and we can see here the cardiac for a mouse weighing approximately 25 grams. The end systolic volume for mouse is normally 25 microliter, but the end systolic volume is approximately the systolic volume is 70 to 80 microliter, but the end systolic volume is 25 to 30 microliter. Micropet imaging action, on the other hand, will provide a powerful functional technique that provide very sensitive measurements of probe concentration. It's a quantitative imaging approach, biokinetics of new diagnostic and treatment agents, stem cell research and gene expression research, cell trafficking and signal transduction, labeled the candidate drug to examine biodistribution used to test the drug pharmacodynamics. and also can combine those other imaging modalities to reveal more information about these characteristics like micro-PET, micro-CT, micro-PET, micro-MR, micro-PET, micro-SPECT, and micro-PET fluorescence. Here, CT and PET and PET-CT, exactly the same feature provided in clinical setting can be applied in preclinical arena. Micro-PET, micro-MR for isochronous for infrared mouse model. Here we can see the infrared between the fused image between the PET and the MR. Here is the timeline for the evolution of micro-PET MR. It started actually in 1997, where some investigators tried to bring the photomultiplier tube outside the magnet. This photomultiplier tube is so sensitive to magnetic field, so the first trial was to bring this photomultiplier outside the magnetic field using optical fibers as light transmitter. And then 2 years or 3 years later, they developed a blanch photodiode. As you can see, it's a blanch photodiode. It is insensitive to magnetic fields, and since that time, researchers started to focus on this blanch photodiode just to combine between PET and MR in the same system. There are some different systems, this system from Cambridge and this system from split different researchers. Actually, they have used a split magnetic field. And at the end, they tried to fit a new system that brought the PET insert inside the magnetic field to acquire a simultaneous PET MR. As a bottom, we can see different applications including neurology and oncology as well. And a lot of applications are emerging in the field. Here, 2 animals, one PET-CT and one PET-MR. We can see the CT signal. Actually, no functional information can be provided, but the PET signal can give you some information about the viability of some tissues and necrotic core, as we heard, as well as here. And these are fused images. For MR, we can find hyper-intense region here. And for PET, we can again look at this necrotic core, and here is the fusion of both. CT can give you some information about the lung tissue, but the MR signal is totally absent. There are different commercial available micro-PET systems. HIDAT is one of the first designs that released into the market, and then CTI and Siemens released different versions, including R4, B4, Focus 120, and Focus 220. And the latest version comes from Siemens, as we mentioned earlier. Just is a tri-modality imaging scanner combined between PET, SPECT, and CT. Explorer Vista come from GE using dual scintillators. Gamma Medica is, as we mentioned, is LYSO and LEGSO scintillators individually coupled of large photodiode detectors. It's a tri-modality system. Here we try to acquire 2 PET tracers at the same time, but the physics of PET actually doesn't allow us to do so. So we started first to inject PK carbon 11 and then FEDG. PK launched actually for 1 hour, and then at the end of the scan we injected FEDG. The 2 images are then fused together with the CT angiography, and here we can see the PK uptake using this white region and the red outline in early frames, but at the end of the scan, completely out of the lung. But FEDG in the heart persists. This actually, the study was conducted to compare between PK carbon 11, 11195, and FEDG in atherosclerosis. These transgenic mice that express atherosclerosis when fed under high cholesterol feeding. And this image actually received a received an award in Kyoto, Japan in 2010. Taking a title feasibility of using more than one imaging biomarker with CT and contrast in the immune system. Optical imaging is 2 classes, bioluminescence and fluorescence. In bioluminescence, it depends on luciferase gene is expressed, and also once you inject into the animal the luciferin, it's being catalyzed in presence of ATP and oxygen to give you light quanta. But fluorescence depends, you inject a fluorescent dye, and the fluorescent dye with the help of external light excitation, it gives you some light photons to give you a biological signal. Optical imaging actually has some good characteristics, like it's a highly sensitive, nano or even femtomolar concentration can be obtained, high throughput imaging technique, easy to use, inexpensive. Optical imagers can be fit in a small size laboratory and also require less space. Limitations of optical imaging are easy to translate from bench to bedside. Some success was recently reported. Limitation of this technique is limited penetration, and near-infrared fluorescence uses 600 to 900 to allow for some photons to come out from the animal. Fluorescence molecular tomography is also one of the options that can improve the quantitation. Here is the difference between fluorescence and bioluminescence. In bioluminescence, you can get a high signal-to-noise ratio, but in fluorescence, there is some tissues or some molecules in the animal can express these phenomena, so the signal-to-noise ratio is much less than bioluminescence. Actually, the system for optical imaging is very simple. It's a small box with CCD camera at the top of the machine, and once you acquire your light photons, it can detect it and give you a biological image signal. Photoacoustic imaging depends on a light source or laser that be absorbed by the light tissue. The light tissue started to expand and give you some ultrasound vibrations. These vibrations can be detected by ultrasound detectors to give you an image. Some applications actually for this modality is in vivo. Some applications for the modality in vivo for tumor angiogenesis monitoring, blood oxygenation mapping, functional brain imaging, and skin melanoma, and etc. These images actually were acquired for a mouse weighing 25 grams. PET-CT were acquired, the FEDG was injected, and CT angiography was acquired after the implementation of the FEDG scanning. We can see here vascular tree, liver vascular tree images, and also the pelvic vascular branches, as is fused together with the bed data. The bed data were histogrammed at different time intervals. We can see the heart very clearly, and also we can see the liver at early frames of the data and the brain at later frames. So the image actually was very multiparametric. In vascular functional imaging in preclinical PET-CT towards more enhanced diagnosis, and this image received actually best image presentation from Siemens in San Diego 2011 and WMIC